for us in the bone marrow, specifically the red bone marrow. So we mentioned bone marrow back in the bone chapter. We talked about red bone marrow, yellow bone marrow. Yellow is mostly made of fat. The red is where we are making new bone cells for us. When you get to embryo, there's a little bit of talk about how you can make our blood cells in the liver and the spleen. So that's stuff that happens the fetus, but as adults, all of our bone cell formation, or blood cell formation, is happening in the bone marrow. So the cells that we're making, we call these myeloid elements. It's just our formed elements. We talked about formed elements in the last chapter, blood being the red blood cells, white blood cells, and platelets. So those are our myeloid elements. Okay, so if you see that word, that's all it means. It's talking about the cells and stuff that we're making for the blood. Cells that we're going to look for. First, we're going to look for reticular cells. These are cells in the bone marrow that make reticular fibers. So that's kind of nicely named for you. In other tissues, we've seen fibroblasts making reticular fibers, but in bone marrow, it's reticular cells. And the predominant fiber type is the reticular fiber, type 3 collagen, so that'd be useful to know. And this is one of our special reticular connective tissues. So we see that there's a lot of reticular fibers in this tissue. So the reticular cells are, again, the ones that make reticular fibers. We also have reticular fat cells. So I think when we looked at bone, I mentioned, we can see fat cells in between the spongy bone within that bone marrow. There's something just kind of helping you figure out where you were. We're actually learning about them now. So the reticular fat cells, they're reticular cells that are filled with fat. They do not come from lipoblasts, so they are not adipocytes. So we're going to see these fat cells. You can call it fat cell. A little more specific, it's the reticular fat cell or fat-filled reticular cell, something. But just as long as you're not calling it adipocyte, that's good. It is still storing triglycerides, but it doesn't really help with metabolism or anything that our normal fat cells help with. So even if you're losing a lot of weight using fat, you're not going to be pulling fat from these cells. They just kind of store it. So again, they're not adipocytes, but we do see fat cells, so we'll just call them fat cells. There are some connective tissue reticular cells that are just providing support for the bone marrow. So you have some that are making the fibers and some that are just almost acting as walls, or forming walls and giving support to the tissue. Macrophages are going to be here. We do have a decent amount of cell turnover in this area, so macrophages can break down any cells that are dying. So we do want macrophages. And then we're going to see tons of developing bone or blood cells. Okay, so that's something we're going to look for. We're not going to identify every cell in bone marrow. A lot of them look very similar to each other and make it almost impossible to identify every cell, especially with the stem cells. We're going to try to figure out which stem cells who. But we will get to see some of the cells as they develop and what they look like. Yeah.
What does hematopoiesis mean?
Blood cell formation. Yeah, so we're making white blood cells and red blood cells in this chapter. Yeah. So when we look at the bone marrow, it is organized. Sometimes it doesn't look super organized, but it is organized into two parts. We have the hematopoietic part that's actually producing new cells. We call those areas cords. And you'll see lots and lots of cells in these cords. And then we have vascular compartments that are made of a special blood vessel called sinusoid. So sinusoid, next on Thursday. Thursday we're going to learn that these are special types of capillaries. So normally you think of capillaries, you think of those really tiny blood vessels that only one blood cell can fit through at a time, help with gas exchange between blood and the tissues. But here, this is a specialized capillary that's huge. It's a giant capillary. But they have very, very thin walls. So they don't have, you're gonna learn that there's layers of blood vessels. Capillaries only have one layer of cells. So we see that here, there's one cell layer thick, we have the endothelium layer. But with the endothelium, it does have a base lamina that's discontinuous. And the reason for the discontinuous base lamina is the endothelial cells actually have big pores, big holes in the cell. And the reason we have those is cells will actually be able to pass from the pores where we're producing new bone or blood cells and pass into the sinusoids. So they don't go in between the cell walls of our sinusoid. They will actually go through the pore in the endothelial cell. So that's how we're getting our new blood cells that we're making in the bone marrow into circulation will actually pass through holes in the endothelium, get into that special capillary called a sinusoid, which will eventually take that blood to the veins of the body, get those cells into circulation for us. So this sort of shows you what that looks like. So the cords are all these areas that are very, very, very cellular. Actually, your bone marrow is amazingly cellular. To say that it's very cellular is an understatement. There's 100 times more cells in the bone marrow than people living in the world. Billions of cells just in the bone marrow. So very, very, very cellular. We're going to see tons of cells when we look at a slide of bone marrow. But most of those cells are going to packed into these cords. So there's going to be cells that helping produce our erythrocytes. We're going to have cells that are helping produce the white blood cells. We have this huge cell called a megakaryocyte helping make what? Yep, platelets.
Those better be granules.
These little things are those.
And inside of the platelets.
Yes, that's the granulomere. Maybe that's what they thought was a nucleus. So people were like, oh, it has a nucleus, but it's just the granulomere.
Yeah.
Maybe. Good thing we know the difference, though, right? Yeah, that's the granulomere. One thing that we might be able to see is usually closer to the sinusoid, so this open areas of sinusoid. On our slide, what you're going to see is that it's packed with red blood cells. So you'll see lots and lots of red blood cells. These areas where we have sinusoids look almost more orange on our slides, I guess. That's what we see with the cords. Fewer cells with nuclei here in the sinusoids. But when you look for a sinusoid, a lot of times what you'll see are cells close by are the ones that are producing red blood cells. So when we talk about red blood cells, they don't have the ability to crawl. So red blood cells can't crawl, they can't migrate through tissue. So if we tried to produce the red blood cells kind of in the middle of a cord, it'd be very difficult to actually get them into circulation. The white blood cells, they do crawl. They have that ability, so we can make them a little further away from the sinusoids, and they can migrate their way once they're mature to circulation. So in general, we usually see these cells making red blood cells closer to the sinusoids. This one they call it a fat cell, but again, this is a reticular cell. It's filled with fat. To be a little more specific, but as long as you don't call it an adipocyte, we're good. Again, the cord's very, very cellular. You're going to see lots and lots of cells. Again, reticulocytes, reticular cells, the fat cells, there's also some fibroblasts somewhere in there. We won't try to identify them, but they're there. Again, cells that are making the peripheral blood cells. So a lot of blood forming cells in this area. So on a microscope, this is what it's going to look like. You'll see some kind of white spaces. Those are the fat cells. The sinusoids, you'll see a little more space and red blood cells. You can still see white blood cells, but again, the majority of them will be the red blood cells. So all these areas that look, I think, a little more orange are the sinusoids. And in between these sinusoids, you see darker staining areas, and these are the cords, where again, we're working on making new blood cells that will eventually make their way to the sinusoids and to the veins. Where we find this stuff is in the red bone marrow. So we find that in between the spongy bone. So that's what this is trying to show. We have the compact bone on the edges, spongy bone inside, and they're just taking a little chunk from the spongy bone and zooming in on the bone marrow between the trabeculae. Down here, there is a cell that they kind of squared around. See how that cell is huge compared to everything around it? That's the megakaryocyte. On your slide, you should be able to find megakaryocytes on the bone marrow slide on your 4X. You shouldn't have to zoom in to find them. You should easily be able to just scan around and find megakaryocytes. Questions about what bone marrow will look like? Yes.
Did you say sinusoid was the capillaries?
Yeah.
Okay.
Yeah, the sinusoid is the special capillary that we're going to see in bone marrow. We'll see sinusoidal capillaries in a few locations, but right now this is our major one. So we'll talk more about those on Thursday. Are you doing that lecture Thursday?
Yeah.
Yeah. So Josh is going to cover the blood vessels with us and talk about the different types of capillaries. All right, so with these blood cell lines, we're first going to talk about how white blood cells form, and we're going to look at the neutrophils specifically. You can see eosinophils and basophils, but the neutrophil is going to be the easiest one to identify, so the only one we'll focus on. And then after that, we'll look at how red blood cells form, and so with the white blood cell line, we're going to start with a cell called a myeloblast. And on, I think, your last page of the notes, give you kind of a chart of how these are, like the flow for each cell as we progress in forming our mature blood cells. So we're going to start, all these start out as coming from stem cells, but we're going to get quickly to the actual cell line. So the myeloblasts are our first cell in line. They can undergo mitosis and give rise to new white blood cells. From there, we're going to get our promyelocyte. On our slides, I don't think really, well, actually we're not even going to try to find a promyelocyte or a myeloblast on our slides. They're difficult to find and distinguish from other cells in the bone marrow. So I'd be aware that myeloblasts are going to give rise to promyelocytes. Both of these can undergo mitosis. But the promyelocyte is then going to differentiate into our granulocytes. So again, the line that we're following is the neutrophil line. So we're going to have a neutrophilic myelocyte that comes up with, we're going to go through each one of these, but we should be able to see myelocytes, metamyelocytes, band cells, or they can also call them stab cells, and then the mature neutrophils. So those are the ones we're going to be able to identify on our slides. So the first one, the myeloblast, we're not identifying that. Just want you to know that that's the first cell in line to make our granulocytes. They undergo mitosis, and that's pretty much it. Myeloblast, first cell in line that we're talking about to make our granulocytes capable of mitosis. That's good. If I ask you what's the next cell in line, the myeloblasts are going to give rise to our promyelocytes. This one will be able to identify the promyelocyte will be able to identify on our slides. What we're gonna see is the promyelocyte is a nice round nucleus. It's usually a little bit more eccentric, so we'll get it off to the side a little bit. So we'll see that with the white blood cells, eccentric nucleus. You might see some nucleoli, but that's not really the thing I'd look for. I'd look for a nucleus that's eccentric and round. These cells, again called promyelocytes, they're capable of mitosis, so they can divide and give rise to the different granulocytes. So this is what you're going to look for. You're going to look for a very large cell in the bone marrow. We have a bone marrow smear slide that we're going to use. Nucleus is round and eccentrically located, kind of squished off to the side. You'll also see some granules in these cells. So that'll help us distinguish if we're looking at a white blood cell line or a red blood cell line. White blood cell line has granules. Red blood cells, we're not going to see the granules. The next cell in line is the myelocyte. You might be able to find some eosinophilic myelocytes, neutrophilic ones. We're going to focus on the neutrophil line. The granules with a kind of neutral pinkish color. But with this one, the nucleus is going to get a little bit smaller, the cell is going to get a little bit smaller, and as we develop these cells in general, the cell line is going to get smaller. The nucleus is going to still be more eccentrically located, but what we're going to start seeing with these myelocytes is the nucleus on one side becomes slightly flattened. And we'll also start to see evidence of a Golgi ghost next to that flat side. So myelocyte, look for eccentric nucleus, flat side on the nucleus for the Golgi ghost. These cells can also undergo mitosis, and this is the last cell in line that can do that. So right there is one. See the nucleus squished off to the side. One side is starting to flatten on the nucleus, and you get a cute little golden ghost white spot next to it. So that is our myelocyte.
The one to the right.
Oh, you can ignore that one. That one's an eosinophilic myelocyte. This one's neutrophilic. We're focusing on the neutrophils. I think a couple more of these have the extra. So when you're looking at it, you're going to start seeing the difference in granules. Remember, the eosinophils have bright red granules. The neutrophils are a little more neutral colored. So that's what we're seeing here is eosinophilic versus neutrophilic. On our slides, I'm only going to test you on neutrophil line. It's a little easier to see since the granules aren't so dark. So like this one, it's hard to tell a Golgi ghost or really a flat side of the nucleus. Where in our neutrophilic ones, everything's just a little easier to see. That's why I'm sticking to that one, just to make it slightly easier. Yeah.
What about the flat side, you say?
Who?
The flat side.
The flat side?
Yeah.
So with the neutrophils, remember they're gonna start lobing out. So this is part of forming those lobes. They start flattening, and then on our next slide we'll see that it'll actually indent in.
Okay.
Yeah, so this is just progressing. So we start to see a flat side, Golgi ghost, so we have the myelocyte. Then our next stage, that nucleus indents even more. That's called a metamyelocyte. These are post-mitotic, so at this point they are starting to mature into the cells that they're going to become. So they can no longer divide, they're just working on maturing into a neutrophil. Again, we have the basophil line, the eosinophil line, neutrophil line. We're focusing on neutrophil. So metamyelocyte will start to indent in. Do we have a way to remember that last M? Starts to look like an M shape, metamyelocyte? I don't know.
I have mnemonics to remember a lot of this stuff,
You do?
but I don't have it with me.
Okay.
So
So in later review dates, we'll give you some memory tricks.
Yeah.
Cool, thanks. You still see a little bit of a Golgi ghost, but we're starting to see an indentation. So this is a metamyelocyte, where the myelocyte was more just flat edge. On your slide, you're going to see all kinds of in-between stages as well. So what I'm going to want you to do is just try to find the one that looks closest to our slide picture. And if it looks somewhere in between, ignore it. You're going to see tons of cells that we're not going to identify. All right, the next cell in line also unable to divide. We call this a band cell, or some folks will call it a staff cell. Either one on here, would you call it? What you're going to see is the nucleus is going to look more U-shaped. So instead of just barely dented, it's a nice U. Again, I don't go for in the middle, somewhere in between a metamyelocyte and a band cell. We're going to pick one that's clearly just slightly indented or a U shape. I'm going to try to do something slightly in the middle of that. Hopefully you can figure it out. That's me. So this one, nucleus, again, is going to look more like a U shape. That's really what's going to help us identify this one. So for U-shaped nucleus. Now, the cell is starting to get smaller, so you're seeing it's getting closer and closer to the size of the red blood cell where we started out. Our first cell was huge. So nucleus is getting smaller, the cell is getting smaller. In general, we're just getting smaller as we're maturing. And then we get to the mature cell, which you guys have learned. I will probably only ask you the mature blood cells on the peripheral blood slide. I'm going to leave the bone marrow slide through those different stages of blood cell development. Right. So there's a nice, pretty neutrophil, what it looks like once it's mature. So that's our stages of what we're going to see with our white blood cell line developing, things that you can see in the bone marrow. And then the red blood cell line, these are our erythrocytes. We're not going to see all of these. So the pro-erythroblasts we will not look for on our slides. It's at those large stages, everything kind of looks the same, and it's hard to distinguish stem cells from pro-erythroblasts, from some of the other cells. So where we're going to start is the basophilic erythroblast. So with our red blood cells, what we're going to see is we do have a nucleus. We start out with the nucleus. It's a pretty large nucleus. Usually it's slightly more centrally located, but sometimes you'll see it squished to the side. But what we're going to see is the nucleus is going to start getting smaller. The cell's going to start getting smaller. And a big thing is the cytoplasm is going to change color. So at the beginning, when we're first forming our red blood cells, remember we want to make a special protein for the function of red blood cells. What protein are we making?
What do you want to do?
Hemoglobin, yeah.
Oh, okay.
Yeah, we want to make hemoglobin. So in the beginning, we're going to have tons and tons of ribosomes. So our cytoplasm is going to look more pale bluish gray color. As our cells maturing and we're becoming closer and closer to a mature red blood cell, our nucleus is going to start to condense and get ready to be spit out. And our cytoplasm, we're going to start decreasing the number of ribosomes and increasing the amount of hemoglobin, which gives more that kind of pinkish red color. So that's what we're looking for in this series, these kind of changes. So in pro-erythroblasts, we're not going to look for this one, but we want to know this is where we're starting our red blood cell line. This cell is going to undergo mitosis and give rise to the basophilic erythroblast. This one, what you're going to look for is a round nucleus, kind of more centered. And the cytoplasm is going to be more of a bluish gray color. And we have lots of ribosomes helping make hemoglobin. So when we look at these cells, get large round nucleus, whatever you want to call that cytoplasm. So that's kind of what it looks like. Nucleus almost takes up the whole space. Yeah. You may even think it almost looks sort of like a lymphocyte, but huge. It's a huge cell, which you'll be able to tell on your slides when we pull up the bone marrow smear. Next up, we're going to have the polychromatophilic erythroblast. This one, what we're going to see is the cell is getting smaller, the nucleus is getting smaller. You have a little more cytoplasm now than we saw in the last one. And this one kind of, to me, looks like the nucleus has sort of a checkerboard pattern. You might just squint at it to see that, but kind of a checkerboard pattern to the nucleus. The cytoplasm is also going to start looking more gray. The last one was more bluish, purpley gray. Now it's looking more palish gray. So the color's starting to change, the cell's getting smaller, we have a checkerboard nucleus. That's what we're looking for on this one. This is our last cell that can undergo mitosis. It's able to make new cells that last in line that can do this. So you're seeing it's getting smaller. You sort of see a checkerboard nucleus, and compared to that last one, the cytoplasm is becoming more pale, starting to resemble a little closer to the color we see with our red blood cells. I have a couple more pictures that I passed out to you that I think do a good job of showing the different stages and helps you compare them, rather than I think sometimes looking slide to slide is a little bit harder. The next one is called an orthochromatophilic erythroblast. At this point, it's also called a normal blast because it's starting to look like a normal red blood cell, except it has a nucleus. So at this point, the nucleus gets really small and dense. Usually you'll see the nucleus more eccentrically located because we're getting ready to spit it out. These are post-mitotic at this point. We're not going to make any new cells. They are what's going to enter circulation once we mature just a little bit more. We're going to spit out that nucleus. Usually this happens in the bone marrow. Every once in a while you'll get one of these out to circulation. So if you see normal blast on your peripheral blood slide, it's okay. But usually not too many of those that you'll see. So this is one we're looking for in the bone marrow slide. So kind of like these. Actually on some of your slides, if you look enough, we're going to see, you'll actually be able to see it spit out the nucleus. You'll see it like partially out and really poking out the side. So the nucleus is getting much more dense, the cell's much smaller, and if you look at the cytoplasm, it's almost the exact same color as our red blood cells, because at this point we are losing the ribosomes and have almost all of the hemoglobin that we need for the cell to do its function. Our next one is a reticulocyte, not to be confused with the reticular cells that make the reticular fibers. It sounds very, very similar. Reticular cell makes reticular fibers. Reticulocyte is in our red blood cell line. This one, we have finally spit out the nucleus. There is a little bit of RNA left, but really not much. It looks almost exactly like a red blood cell at this point. To tell them it's a reticulocyte, we actually need a special stain, and we don't have that. So we're never going to identify a reticulocyte. I would know that it's the last stage before we become a erythrocyte. I would know that it's in this red blood cell line. If we tag a orthochromatophilic or normal blast cell and ask you what's the next stage that you could say it becomes a reticulocyte, but you won't have to identify a reticulocyte. With this special stain, you can pick up some of that leftover RNA, but we don't have that, so we're not going to identify And then we have our mature red blood cell. And that's definitely something that is going to, or could be on the exam.
I remember one mnemonic.
Yeah.
This erythrocyte one. If you take the first letter of each of the words that are through this process, it's like PBPORE, PB4. So a way to remember that, for some reason, I thought it peanut butter pork.
Peanut butter bowl.
So that I could use that and list the first letters and then write out in order which one was which.
Yeah, that works. The next one's rough because you have so many M's.
Yeah. I think I have one for that too, but I can't remember. That was just the one I remembered off the top of my head.
Oh, okay. I like it. Alright. Our platelet line. All I want you to know is megakaryocytes give rise to our platelets. So the megakaryocyte we're going to see is a bone marrow. It's huge, has a huge nucleus, and again, it's going to kind of just fragment off and give rise to the platelets. And again, they're huge. We'll be able to identify them easily. And hopefully we'll even be able to see where platelets are piecing off on some of them. Again, the whole cell line for those, just know megakaryocyte to platelets. That's it. And then we are not going to do the lymphocyte, monocyte lineage. They come from different cells than our granulocytes. You can see a flow chart on the last page. It shows you where the monocytes are coming from, shows you where the lymphocytes are coming from, but we're not going to do that. So on that last page with the chart, pretty much you need to know the proerythroblast down to erythrocyte. You need to know megakaryocyte to platelets, and then myeloblast and just that neutrophil side. And you already know monocytes become macrophages, but the whole cell line for the lymphocytes and monocytes, we're not going to. One thing that I maybe would note is these cells don't just multiply and make new cells out of nowhere. There are a lot of growth factors that stimulate cell production. So it's not like your red blood cells just start producing more red blood cells just to do it. They're going to respond to erythropoietin, which actually comes from the kidneys. There's a lot of other stimulating factors for producing new cells and getting them to go through mitosis. Just to keep that in mind. I'm not going to ask you the specifics of that. Just growth factors help with this. Yeah.
So for the which one did you say that we aren't doing?
Sorry, I can't hear you.
For the flow chart, which one did you say that we're not doing?
That we're not doing. We're not doing the monocyte, so you don't have to know monoblast or monocyte. The lymphoid restricted stem cells, we don't have to go through the lymphoblast to the large lymphocytes, to the medium to the small. We're not doing that line. For the myeloblast to promyelocyte, we're only doing the neutrophil side. And then the platelet list, we're only doing megakaryocyte to platelet. Is that here? Label some of this stuff real quick with you guys. Here's what I got. nobody was like taking pictures during class. Things changed so much. Alright, so real quickly, yeah.
Pulls that camera.
Yeah. Yeah. Can you get my camera out real quick? Let me see if it has enough memory left. I was an old.
See if I can film you.
Gotta go to CBS to develop.
Yeah. Hope that works. Oh gosh. Man, things changed. Alright, so with this, I thought we could maybe just really quick make some notes on here of some important things. So again, we're not going to look for the erythroblast. This is the red blood cell line on your chart. I gave you asterisks next to the ones that we will be able to identify on the bone marrow slide. Okay, so the asterisks mean we're looking for it on the bone marrow slide. The line here and here means this is our division between what can undergo mitosis, what can't. So with our red blood cell line on top, the proerythroblast all the way to the polychromatophilic erythroblast, these can undergo mitosis, these cannot. Then down here we have the white blood cell line, and the asterisks mean those are the ones we're going to identify on our bone marrow slides. Here's our line, so these two are mitotic cells. Then once we get to the metamyelocyte, we're no longer able to undergo mitosis. So that's what those mean. Yeah, we're not going to look for the proerythroblast, but we will look for a basophilic erythroblast. So this one, you want the kind of blue cytoplasm. And this one has more of a round centered nucleus. Then the polychromatophilic, when you're looking on your slides, and this one sort of has a checkerboard nucleus. And the cytoplasm is starting to become more like a pale gray cytoplasm. At this point, we're saying really decrease the number of ribosomes and increasing the amount of hemoglobin. So that's why we're seeing this color change. By the time we get to the normoblast, it has more of a normal red blood cell cytoplasm color. Small, dark nucleus, which is more eccentric. If normal color cytoplasm is not good enough, you can call it maybe like salmon pink cytoplasm color. And then erythrocytes are just the normal red blood cells. We're not looking for reticulocytes. I would know that reticulocytes are here in the line. But we're not going to look for them, so there's no picture of them. White blood cell line, again, we're looking for the neutrophil line. Promyelocyte, you want a round nucleus. It's located eccentrically. And we're going to look for kind of pinkish granules. No granules in our red blood cell line. They're not secretory cells. They do not need granules. White blood cells can do secretory, they do secretion, so we have secretory granules. So look for pink granules. And then for the myelocyte, we start to see this kind of flatter edge on the nucleus. So flat side nucleus. You can see right here the bulge goes. and the nucleus is more eccentrically located. Our metamyelocyte, I'm gonna look for indented nucleus. You don't see any indentations, everything stays pretty round with our erythrocyte line. With our white blood cells, you see a metamyelocyte has an indentation. Then this one we will look for a U-shaped nucleus. And the neutrophil will identify that on our peripheral blood slide. The lab is one that we usually don't do together because the quality on our screen is awful. So it's one that usually I have you guys kind of work on your own first, and then maybe take pictures and ask questions about what you're looking for, and something that usually you cover more in a review session. So what I did was print out exactly what I want you to look for on the slides. So your first slide is box 1, slide 87. That's a peripheral blood slide. It's a special stain for blood. It's the right stain. Nothing to do with right state, but I like it anyways. So this one, you're looking for red blood cells and all 5 white blood cells if you can find them, and then the platelets. Box 1, slide 88. We are looking for the just normal bone marrow, so it's a section of bone marrow. You're going to look for the cords. The cords are where you're going to find the fat-filled reticular cells. You'll find megakaryocytes here and developing blood cells. They'll be very difficult to distinguish the different types of developing blood cells on this slide. So just look for the difference between cords and sinusoids, basically, and some megakaryocytes. The sinusoids, they'll look a little more kind of orangish red, because again, we're going to have a lot of the mature red blood cells in those areas. And then box 1, slide 89, that's where you're going to try to find the cells that are part of the red blood cell line and white blood cell line. You may be able to find a mature plasma cell. Just because you already know what those are, we might as well look for them. And then answer some questions underneath that, just to be kind of focused on the importance of the lab. So we can hang around for a little while if you want to do that now, or you prefer to try to find them first on your own and then ask them on the review session. We don't have to skip today. That's fine. We're basically out of time anyways. So if you are hanging out here and want to study and try to look for them, I'm going to be here for a little while as you to prepare for. So I'll be here. You said you're sticking out after class?
Yeah, if anybody wants to go over questions.
Yeah, so if you want help with it, I recommend sticking around doing it while someone's here to help you. But if you want to do it on your own, that's completely fine too. Just definitely take the time, practice identifying them, and try to find ones that look like the pictures in the notes. If you see cells that don't look like pictures in the notes, don't try to identify it, just skip it. Because if you try to identify every cell you see on the slide, you will go crazy. At least I would. My opinion, I guess. But yeah, so I would practice first, and then maybe on Thursday after class we can practice again. After you guys have tried to look for them on your own. But definitely use those pictures in the packet that I have handed out. Do you have any questions? Holler if you do. And also, I'll be on campus a lot this week if you guys have questions.
What happens if you're already crazy?
If you're already crazy,
Yeah.
I might not get this.
You still be
find
driven
out.
crazy if you're already.
Can you go crazier?
That's true.
I have a psychology question.
Depends on what category you're crazy.
Maybe.
Always fun.